You are out of place for my talk, but I hope I'll give you some interesting food for thought, good food. I just got a message from Noor, our friend who is missing all of us in this meeting, and he insisted that I give this type of talk. So, information highways in cells and tissues. I should say initially that I'm an engineer. I studied engineering, chemical engineering, then worked in the States in the development of the first artificial heart, and that made me fall in love with biology, and this love is expanding instead of diminishing. So, what am I going to tell you here? That doesn't work, so yeah. Everybody makes a disclosure, so here I copy Socrates saying that true wisdom comes to each of us when we realize how little we understand about life, about ourselves, and about the world around us. So we are still in the infancy of understanding what is happening in our bodies. So, information. What is information? Is it knowledge? Is it a component of knowledge? Is it always a fact? Is it a signal only, a message? What is it about misinformation? Sometimes in a transcription, there is misinformation. Or a tumor cell might send disinformation into the body. They communicate all things around in the body. They cohabitate. There are opposite or different sources of information. They fight among each other, or they collaborate. So competition and cooperation is always in a dynamic state in our bodies. Now, before I say a few more things, let me show you some numbers that boggle my mind and probably many others. So, what are people? People are a superorganism with human and bacterial cells, along with viruses that co-inhabit, antagonize, fight or protect each other. And the numbers there, you see we have 10 to 12 cells, somatic cells, but many more bacteria and viruses. So if you think about neurons, we have numbers of the 10 to 10th neurons in the body. If we need to talk about flow of information by entities like proteins or something, we have to think about viscosity. Things are not flow like water in our body. We think it is, but we have to know the viscosity, which is 5 times that amount that we know of water in certain places. Number of cell divisions in the lifespan of us, 10 to 16, huge numbers. Blood cell turnover, every day we have 100 million blood cell turnover. So the numbers are big. Now, what is happening in a cell? I will show you a video which is not a real cell, but very, very close to reality. And look what is happening. Can you put sound, please?
Throughout the cytoplasm, forming the struts, cables, and girders that give the cell its shape, internal organization, and mechanical support for movement. There are several types of protein filament, each built from subunits that rapidly come together or
real time,
allowing these elaborate structures to be assembled and dismantled wherever they're required by the cell. Microtubules are stiff, hollow tubes that anchor other organelles and serve as tracks that guide the movement of vesicles and other cell components. actin are thin, flexible filaments that form cross-linked bundles and branching networks. Actin is particularly important for cell movement. They are central to the contractile engine in muscle cells and provide the mechanical force for cell movement at the plasma membrane.
So, there is chaos, huge numbers, different time scales of things happening, information is being translated, sent to each other, and how we are going to study this. What is the methodology we follow in science? At least in Greek universities or US universities, we do not learn much about methodology. We just follow. So the question is science progressing by accumulating new established truths upon old ones? Is it a built-up thing? Or, like other people have suggested, the process is not cumulative or continuous. Earlier results may be rejected, replaced, and reinterpreted by new theories and conceptual frameworks. Now, to go and study little things, we have to use reductionism. We take the whole and we go down. Relative isolationism of systems is investigated, but we lose information when we take a cell out of the billions of cells to study it. We have to use a dialectical approach or a holistic approach, whatever term you like, that gives us contradictions, negations of negations, synthesis of antithetic positions, etc. And in this sense, biomimetics or reverse biomimetics is useful. Now, if you have something moving in the body, say a protein or a nanoparticle, there are at least 3 different ways that they move around. Why they move? To connect, to give information. One is diffusion, so we have to know the diffusion coefficient of a substance, or motor-driven, like you saw in actinomycin, or advection. And these are not isolated. Sometimes a protein follows both diffusion, but also advection. So already the complexity becomes more complex. Now, a few years ago, 5 years ago, I chose a term which was published called mechanoepigenetics. What that says is this, it says that if we have a cell, the cell is connected to other cells and flows around it, and also communication with the extracellular matrix. Now, all these kinds of signals come into the cytoplasm. They move like you saw in the cartoon video, and eventually they enter the link, through the link complex, into the nucleus, through the lamins, and go to the chromatin. Now, if you know that chromatin is so entangled that nothing can penetrate, so mechanics is very important to go there first, disentangle chromatin, and then have genetic expression. Otherwise, we die. So, information, how we send this information is important and very little understood. So, again, to summarize this one, basic modes of communication in the body. There is direct cell-to-cell, including gap junction cell surface protein interaction. There is an extracellular matrix nucleus communication via mechanotransduction. And with distant cells, we have hormones, cytokines, chemical messengers, mechanical electrical signals. And one subject that has attracted lots of attention in this sense is the tumor immune cell communication. Now, recently, a lot of studies are done on exosomes. What are exosomes? There are about 1 million exosomes per milliliter in plasma, and each one has about 500 microRNAs. Now, just think about it. With the trillions of exosomes moving around, good ones or bad ones from cancerous cells and others from benign cells, and so on and so forth, carrying all this information in their microRNAs. Why they do it? How they do it? Very little is known. But it's very important. If we understand a little bit of that, then we can use it for prevention instead of later on needing some of you guys here. They are found in many body fluids. They contain, apart from microRNAs, DNA, proteins, lipids. They produced almost by every cell. They travel and they are taken by other cells. How they recognize? Is there redundancy in nature? These are questions that we ask, but still not systematic study about it. They are implicated in cancer detection, prognosis, and therapy. For example, when you have hypoxia, more tumor exosomes are released. They proliferate. There is proliferation of endothelial cells, and then there is immune suppression. Now, if you take, I'm not going to analyze this slide, but if you have a tumor cells, it communicates with many other cells, including neutrophils, NK cells, dendritic cells, and so on, through exosomes apart from other information. So you see, one cancerous cell has so many exosomes that he sends away and receives. And this goes on all the time. Homeostasis. Now, I'll go very fast because the question is, we have to have information security, coming back to information. There are computer viruses in real life. There is encryption. There is biosteganography. There is molecular cryptography. So, in life, do we have to secure the information that goes on through exosomes and other signals? Well, there are genetic, epigenetic, transcriptomic, proteomic variations. There is a huge number of interacting players. So the body needs its own password. What is a password in biology? The right combination of growth factors and tumor factors for reprogramming. Firstly, to access the inaccessible chromatin sites. Stem cells need more complex password to avoid becoming cancerous. So this is reverse biomedics, as I call it. I'm not going to continue on this. Okay, and I end up, because maybe this is a little bit out of place, this talk, again with a Greek poet, since you are in Athens. He got a Nobel Prize, Odysseus Elitis, and in one of his poems, he says, in αυτός ο κόσμος ο μικρός ο μέγας, in Greek, which means, translated by me, the small world, the great, that if you decompose Greece, you will see that what remains is an olive tree, a vineyard, and a boat. This means that with these three ones, you can reconstruct it again. And the poem is in Mikros Naftilos. Now, poets can do whatever they like. They have the right given by society to say nice things. If you disassemble an organism in order to examine the way its awesome parts function, how can you reassemble it and ascertain its function based on the dynamics of its parts? I leave you with this question, my friends. Thank you very much.